61 Chemical regulation of respiration. The ultimate goal of respiration is to maintain proper concentrations of O2, CO2 and hydrogen ions in the tissues. It is fortunate therefore that respiratory activity is highly responsive to changes in each of these substances. Excess CO2 or excess H plus ions in the blood mainly act directly on the respiratory center itself, causing greatly increased strength of both the respiratory and inspiratory and expiratory motor signals to the respiratory muscles. Oxygen, in contrast, it acts more on the peripheral chemoreceptors. It does not have a significant direct effect on the respiratory center of the brain in controlling respiration. Instead, it almost acts entirely on the peripheral chemoreceptors located in the carotid and aortic bodies, and these chemoreceptors in turn transmit appropriate nervous signals to the respiratory center for control of respiration. Now, coming to the direct chemical control of respiratory center activity by CO2 and H plus ions, the chemosensitive area of the respiratory center is beneath the ventral surface of the medulla. There are mainly three areas of the respiratory center: the dorsal respiratory group or DRG, the ventral respiratory group or VRG, and the pneumotaxic center in the pons. It is believed that none of these is affected directly by changes in blood CO2 concentration or H plus concentration. Instead, an additional neuronal area called the chemosensitive area, it is located bilaterally only 0.2 mm beneath the ventral surface of the medulla. This chemosensitive area is highly sensitive to changes in either blood PCO2 or H plus ion concentration, and it in turn excites the other portions of the respiratory center. Excitation of the chemosensitive neurons by H plus ions is likely the primary neurons in the chemosensitive area are especially excited by H plus ions. In fact, it is believed that H plus ions may be the only important direct stimulus for these neurons. However, H plus ions do not easily cross the blood-brain barrier. For this reason, changes in H plus concentration in the blood have considerably less effect in stimulating the chemosensitive neurons than do changes in the blood CO2, even though CO2 is believed to stimulate these neurons secondarily by changing the H plus ion concentration as explained now. CO2 stimulates the chemosensitive area. Although it has little direct effect in stimulating the neurons in the chemosensitive area on its own, it does have a potent indirect effect. It has this effect by reacting with the water of the tissues to form carbonic acid. CO2, when it is in excess in the blood, it reacts with water, CO2 plus H2 forms H2CO3, which passes through the blood-brain barrier, dissociates into H plus and HCO3 minus, and these indirectly synthesized H plus ions have a potent direct stimulatory effect on the respiration. Okay, you can see these reactions here. CO2 in the blood, it combines with water to form H2CO3, which can cross the blood-brain barrier. However, H plus ions in the blood cannot cross the blood-brain barrier. So CO2 has an indirect effect on the chemosensitive area, and H plus has a direct effect on the chemosensitive area. H plus that is produced by CO2 has a direct effect. Okay, so this is shown here. So the blood-brain barrier is not very permeable to H plus ions, but CO2 passes through this barrier almost as if the barrier did not exist. Consequently, whenever the blood PCO2 increases, so does the PCO2 of both the interstitial fluid of the medulla and the cerebrospinal fluid. In both these fluids, the CO2 immediately reacts with the water to form new hydrogen ions. Thus, paradoxically, more H plus ions are released into the respiratory chemosensitive area of the medulla when the blood CO2 concentration increases than when the blood hydrogen ion concentration increases. For this reason, respiratory center activity is increased very strongly by changes in blood carbon dioxide. Decreased stimulatory effect of CO2 after the first 1 or 2 days. Excitation of the respiratory center by CO2 is great the first few hours after the blood CO2 first increases, but then it gradually declines over the next 1 to 2 days, decreasing to about 1/5 the initial effect. Part of this decline results from renal adjustment of the H plus ion concentration in the circulating blood back towards the normal after the CO2 first increases the H plus concentration. The kidneys achieve this readjustment by increasing the blood bicarbonate, which binds with the hydrogen ions in the blood and CSF to reduce their concentrations. But even more importantly, over a period of hours, the bicarbonate ions also slowly diffuse through the blood-brain and blood CSF barriers and combine directly with the H plus ions adjacent to the respiratory neurons as well, thus reducing the H plus ions back to the normal. So that is why the kidneys compensate by increasing the absorption of HCO3 minus ions. These HCO3 minus ions just slowly diffuse through the blood-brain barrier and combine with the H plus ions formed by the carbon dioxide, and then they bring the concentrations back to normal. A change in the blood CO2 concentration therefore has a potent acute effect on controlling respiratory drive, but only a weak chronic effect after a few days adaptation. Okay, so here you can see what happens When arterial CO2 increases, CO2 in the ECF increases. It combines with water to form HCO3, which dissociates into H plus and HCO3 minus ions. Okay, H plus ions increase, so this increases the rate of firing of the chemosensitive areas. And also, if there is an increase in the arterial oxygen content, it stimulates the peripheral chemoreceptors in the carotid and aortic bodies, and this causes an increase in the firing of inspiratory centers, motor neurons to the diaphragm, and external intercostal muscles, that is the inspiratory muscles, they increase the firing rate, and finally the diaphragm contracts, the EICM, the external intercostals contract, and this increases the ventilation. Okay. Coming to the quantitative effects of blood PCO2 and hydrogen ion concentration on alveolar ventilation, in this figure you can see the quantitatively the approximate effects of blood CO2 and blood pH on alveolar ventilation. So note especially the very marked increase in ventilation caused by an increase in PCO2 in the normal range, okay, between 35 to 75 mm of Hg, which demonstrates the tremendous effect that the CO2 changes have in controlling respiration. Okay, there is a tremendous increase in ventilation caused by an increase in PCO2 in the normal range. By contrast, the change in respiration of the normal blood pH range, which is between 7.3 and 7.5, is less than 1/10 as great. Okay, so change in the PCO2 causes a change in the minute change in the PCO2 causes great change in the alveolar ventilation, whereas large changes in the pH only cause little changes in the alveolar ventilation. Okay, this is what is shown here. Changes in O2 have little direct effect on control of the respiratory center. Changes in O2 concentration have virtually no direct effect on the respiratory center itself to alter its respiratory drive. Although O2 changes do have an indirect effect acting through the peripheral chemoreceptors. We learned earlier that the hemoglobin oxygen buffer system delivers almost exactly normal amounts of O2 to the tissues even when the pulmonary PO2 changes from a low value to as low as 60 mm of Hg up to a high value as much as 1000 mm of Hg. Therefore, except under special conditions, adequate delivery of oxygen can occur despite changes in lung ventilation ranging from slightly below 1/2 the normal to as high as 20 or more times the normal. This is not true for CO2 because both the blood and tissue PCO2 change inversely with the rate of pulmonary ventilation. Thus, the processes of animal evolution have made CO2 the major controller of respiration, not O2. Yet for those special conditions in which the tissues get into trouble for lack of O2, the body has a special mechanism for respiratory control located in the peripheral chemoreceptors outside the brain respiratory center. This mechanism responds when the blood oxygen falls too low, mainly below a PO2 of 70 mm of Hg. Coming to the peripheral chemoreceptor system for control of respiratory activity and the role of oxygen in respiratory control. In addition to control respiratory activity by the respiratory center itself, still another mechanism is available for controlling respiration, and this is the peripheral chemoreceptor system, which is shown in this figure. Okay, special nervous chemical receptors called chemoreceptors are located in several areas outside the brain. They are especially important for detecting changes in oxygen in the blood, although they correspond to a lesser extent to changes in CO2 and H plus ion concentrations. The chemoreceptors transmit nervous signals to the respiratory center in the brain to help regulate respiratory activity. Most of the chemoreceptors are in the carotid bodies. C carotid. So most of them are in carotid bodies. A few are also in the aortic bodies, and a very few are located elsewhere in association with other arteries of the thoracic and abdominal regions. Okay, the carotid bodies are located bilaterally in the bifurcations of the common carotid arteries into internal and external carotid arteries. Their afferent nerve fibers pass through the herring nerves, as you can see here. This is the herring nerve. They pass through the herring nerves to the glossopharyngeal nerve, and then to the DRG of the medulla. Okay. The aortic bodies are located along the arch of the aorta, and see, because the aortic bodies are located lower down, they require a long cranial nerve, which is a vagus nerve. The afferent is a vagus nerve. So the afferent nerve fibers pass through the vagi also to the dorsal respiratory area. Each of the chemoreceptor bodies receives its own special blood supply through a minute artery direct from the adjacent arterial trunk. Further, blood flow through these bodies is extreme, 20 times the weight of the bodies themselves each minute. Therefore, the percentage of oxygen removed from the flowing blood is virtually zero. This means that the chemoreceptors are exposed at all times to arterial blood and not venous blood, and their PO2 values are arterial PO2 values. Coming to decreased arterial oxygen, it stimulates the chemoreceptors. When the oxygen concentration in the arterial blood falls below the normal, the chemoreceptors become strongly stimulated. You can see this effect in this figure, which shows the effect of different levels of arterial PO2 on the rate of nerve impulse. As the arterial PO2 keeps decreasing, the rate of nerve impulse keeps increasing, especially when it falls below the level of 60, below the level of 60. From 60 to 30, it is especially sensitive, okay? Because this is the range in which hemoglobin saturation with oxygen decreases rapidly. Increased CO2 and H plus ion concentration stimulates the chemoreceptors. An increase in either CO2 or H plus ion concentration also excites the chemoreceptors, and in this way indirectly increases respiratory activity. However, the direct effects of both these factors in the respiratory center are much more powerful than their effects mediated through the chemoreceptors. Yet there is a difference between the peripheral and central effects of CO2. The stimulation by way of the peripheral chemoreceptors occurs as much as 5 times as rapidly as central stimulation. So the peripheral chemoreceptors might be especially important in increasing the rapidity of response to CO2 at the onset of exercise. So that is why peripheral chemoreceptors act immediately and central chemoreceptors take some time to act. Okay, coming to differences between the central and peripheral chemoreceptors. See, the location, central are located centrally in the medulla, and the carotid and aortic bodies constitute the peripheral chemoreceptors. Stimuli for the central is increase in blood CO2 levels, which causes an increase in H plus ions, and peripheral chemoreceptors are stimulated mainly by decrease in arterial oxygen content, and to a lesser extent by increase in CO2 and H plus ions in the blood. Rapidity, the rapidity in response, the central chemoreceptors are slower, and peripheral chemoreceptors are 5 times as rapid as central stimulation. The exact means by which low PO2 excites the nerve endings in the carotid and aortic bodies are still not completely understood. However, these bodies have multiple high characteristic glandular cells called glomus cells that synapse directly or indirectly with the nerve endings. Current evidence suggests that these glomus cells function as the chemoreceptors and then stimulate the nerve endings. Glomus cells, which are in the peripheral chemoreceptors, they have oxygen sensitive potassium channels, O2 sensitive K plus channels, that are inactivated when the blood PO2 decreases markedly. Okay, so when the blood PO2 decreases markedly, this causes the O2 sensitive K plus channels to become inactivated. Because the O2 sensitive K plus channels are inactivated, there is an increase in the intracellular potassium content, which causes depolarization, and this voltage difference causes calcium channels to open. Voltage gated calcium channels then open, and there is an increase in the influx of calcium into the cell. This extra calcium, this increased calcium ions, they stimulate the release of a neurotransmitter that activates efferent neurons that send signals to the CNS and stimulates respiration. Okay, so although early studies suggested that dopamine, mostly dopamine or acetylcholine, might be the main neurotransmitters, more recent studies suggest that during hypoxia, adenosine triphosphate ATP may be the key excitatory neurotransmitter released by the carotid body glomus cells. Okay. Coming to the effect of low arterial PO2 to stimulate alveolar ventilation when arterial CO2 and H plus ions remain normal. Only the ventilatory drive. See, you can see this figure. Okay. This shows the effect of low arterial PO2 and alveolar ventilation when the PCO2 and H plus ion concentrations are kept normal and constant at their normal levels. Okay, so in other words, this figure shows that only the ventilatory drive, because of the effect of low O2 on the chemoreceptors, is active. This figure shows that almost there is no effect on ventilation. As you can see here, there is no effect on ventilation as long as the arterial PO2 remains higher than 100 mm of Hg. Okay, and there is no, as long as the arterial PO2 remains higher than 100 mm of Hg, there is no effect on alveolar ventilation. However, at pressures lower than 100 mm of Hg, as you can see, lower than 100 mm of Hg, ventilation approximately doubles when the arterial PO2 falls to 60, and then it increases as much as 5-fold at very low PO2 values. Under these conditions, low arterial PO2 obviously drives the ventilatory processes quite strongly. Because of the effect of hypoxia on ventilation is modest for PO2 values greater than 60 to 80 mm of Hg, the PCO2 and H plus ion response are mainly responsible for regulating ventilation in healthy humans at sea level. Okay, so that is what is shown here. Chronic breathing of low oxygen stimulates respiration even more, and this is the phenomenon of acclimatization. Mountain climbers have found that when they ascend a mountain slowly over a period of days rather than in a period of hours, they breathe much more deeply and therefore can withstand far lower atmospheric O2 concentrations than when they ascend rapidly. This is called acclimatization. The reason for acclimatization is that within 2 to 3 days, the respiratory center in the brainstem loses about 4/5 of its sensitivity to changes in PCO2 and H plus ion concentration. Therefore, the excess ventilatory blow off of CO2 that would normally inhibit an increase in respiration fails to occur, and low oxygen can drive the respiratory system to a much higher level of alveolar ventilation than under acute conditions. Instead of the 70% increase in ventilation that might occur after acute exposure to O2, low O2, the alveolar ventilation often increases 400 to 500% after 2 to 3 days of low O2, which helps immensely in supplying additional oxygen to the mountain climber. Coming to the composite effects of PCO2, pH, and PCO2 on alveolar ventilation, this figure, it gives us a quick overview of the manner in which the chemical factors PO2, PCO2 and pH together affect the alveolar ventilation. See, to understand this diagram, we need to first observe the 4 red curves. Okay, these curves are recorded at different levels of arterial PO2 at 40, 50, 60 and 100 mm of Hg, as you can see here. Okay, for each of these curves, the PCO2 was changed from lower to higher levels. Okay, thus this family of red curves, it represents the combined effects of alveolar PCO2 and PO2 on ventilation. Okay, now observe the green curves. Whereas the red curves were measured at a blood pH of 7.4, the green curves were measured at a pH of 7.3. Okay, we now have two families of red and green curves representing the combined effects of PCO2 and PO2 on ventilation at two different pH values. Still, other families of curves would be displaced to the right at higher pHs and displaced to the left at lower pHs. So that is why we can use this diagram to predict the level of alveolar ventilation for most combinations of alveolar PCO2, alveolar PO2 and arterial pH. Okay, so this basically says that when you have PO2 and PCO2 at fixed levels, the graph is somewhat like this, but whenever you decrease the pH, there is a left shift in the same graph, same curves. Okay, now coming to the regulation of respiration during exercise. During strenuous exercise, O2 consumption and CO2 formation can increase as much as 20-fold. Yet in the healthy athlete, alveolar ventilation ordinarily increases almost exactly in step with the increased level of O2 metabolism. The arterial PO2, PCO2 and pH remain almost exactly normal. Okay, so what causes this intense ventilation during exercise? At least one effect seems to be predominant. The brain, on transmitting motor impulses to the exercising muscles, is believed to transmit collateral impulses to the brainstem at the same time to excite the respiratory center. This action is analogous to the stimulation of the vasomotor center in the brainstem during exercise that causes a simultaneous increase in arterial pressure. Actually, when a person begins to exercise, a large share of the total increase in ventilation begins immediately on initiation of the exercise, before any blood chemicals have had time to change. It is likely that most of the increase in respiration occurs from neurogenic signals transmitted directly into the brainstem respiratory center at the same time that signals go into the body muscles to cause muscle contraction. Coming to interrelation between chemical factors and nervous factors in the control of respiration during exercise, when a person exercises, direct nervous signals presumably stimulate the respiratory center almost the proper amount to supply the extra O2 required for exercise and to blow off the extra CO2. Occasionally, however, the nervous respiratory control signals are either too strong or too weak. Chemical factors then play a significant role in bringing about the final adjustment of respiration required to keep the O2, CO2 and H plus ion concentrations of the body nearly as normal as possible. Okay, this process is demonstrated in this figure. So, note that on the onset of exercise, the lower curve, it shows changes in alveolar ventilation during a 1 minute period of exercise, and the upper curve shows the changes in arterial PCO2. Note that at the onset of exercise, the alveolar ventilation increases almost instantaneously. Okay, alveolar ventilation increases almost instantaneously. As much as you start increasing your respiratory rate, it increases. Okay, this increase in ventilation is usually great enough so that it first actually decreases the arterial PCO2 below the normal. You hyperventilate, and this causes washout of the CO2 that is built up in the body. Okay, the presumed reason is that the ventilation forges ahead of the buildup of blood CO2 is that the brain provides an anticipatory stimulation of respiration at the onset of exercise, causing extra-alveolar ventilation even before it is necessary. However, after about 30 to 40 seconds, the amount of CO2 released into the blood from the active muscles, okay, it also causes an increase in the blood PCO2. Okay, and it approximately matches this increased rate of ventilation, as you can see here. Okay, and the arterial PCO2 returns essentially to normal even as the exercise continues. Okay, now in this figure you can see the summary of control of respiration during exercise in another way, this time more quantitatively. The lower curve of this figure shows the effect of different levels of arterial PCO2 on alveolar ventilation when the body is at rest, that is, it is not exercising. Okay, the upper curve shows the approximate shift of this ventilatory curve caused by neurogenic drive from the respiratory center that occurs during heavy exercise. Okay, so the points indicated on the two curves show the arterial PCO2 first in the resting state and then in the exercising state. Note in both instances that the PCO2 is at the normal level of 40 mm of Hg. In other words, the neurogenic factor shifts the curve almost about 20-fold in the upward direction, so ventilation almost matches the rate of CO2 release, thus keeping arterial PCO2 near its normal value. So this also shows that if during exercise the arterial PCO2 does change from the normal value of 40, it has an extra stimulatory effect on ventilation at PCO2 value greater than 40 mm of Hg and a depressant effect at a PCO2 value less than 40 mm of Hg. Okay, in this figure you can see the various factors that regulate ventilation during exercise. Okay, collateral motor impulses to the muscles and the respiratory centers from the brain, proprioceptors and mechanoreceptors in the limbs, chemoreceptors both central and peripheral, brain's learned response, and also your temperature receptors, your nociceptors and all that. See, when your brain is sending impulses to the motor impulses to the muscles, it also sends collateral impulses. Okay, collateral impulses. So this increases, these go to the DRG, that is the nucleus of tractus solitarius, and increase the firing rate. They cause an inspiratory ramp signal. Okay, this signal is transmitted to the inspiratory somatic neurons, and this causes a contraction of the diaphragm and EICM, causing inspiration. Okay, when the impulses reach the VRG, that is when there is pain, when the nociceptors they sense pain, or when the cardiac receptors cannot bear any more exercise, they send impulses to the ventral respiratory group of neurons, VRG, and the brain's learned response to only tolerate some amount of increased ventilation. They reach the VRG, and then these impulses they pass on to the somatic neurons, and this causes contraction of the internal intercostal and abdominal muscles, which causes expiration. Okay, this is what is shown here. Neurogenic control of ventilation during exercise may be partly a learned response. Many experiments suggest that the brain's ability to shift the ventilatory response curve during exercise, as shown here in this figure, okay, is at least partly a learned response. That is, with repeated periods of exercise, the brain becomes progressively more able to provide the proper signals required to keep the blood PCO2 at its normal level. Also, there is reason to believe that even the cerebral cortex is involved in this learning, because experiments that block only the cortex also block the learned response.